Hesi A2 Biology Practice Test. Question 1. In cellular respiration, the molecule that enters the Krebs cycle after combining with oxaloacetate is what? Glucose, pyruvate, acetyl-CoA, citrate. Acetyl-CoA, 2C, combines with oxaloacetate, 4C, to form citrate, 6C. Glucose is broken down earlier, pyruvate is converted to acetyl-CoA, citrate is the first product. Question 2. The specific sequence of 3 nucleotides on mRNA that codes for an amino acid is called a what? Anticodon, codon, exon, promoter. Codon on mRNA pairs with anticodon on tRNA. Exon is coding DNA, promoter initiates transcription. Question 3. Which type of microscope would be most suitable for viewing the internal structure of a living cell? Scanning electron microscope, transmission electron microscope, phase contrast microscope, atomic force microscope. Phase contrast microscopes observe live cells without staining. SEM and TEM require fixed/dead samples. AFM scans surfaces, not internal live structures. Question 4. A cell that lacks a nucleus and membrane-bound organelles is classified as what? Eukaryote, prokaryote, protist, fungi. Prokaryotes, bacteria/archaea, lack nucleus and membrane-bound organelles. Eukaryotes have them. Protists and fungi are eukaryotes. Question 5. The process by which plants convert light energy into chemical energy, glucose, is known as what? Chemosynthesis, cellular respiration, photosynthesis, fermentation. Photosynthesis uses light to make glucose. Chemosynthesis uses chemicals, cellular respiration breaks glucose, fermentation is anaerobic ADP production. Question 6. Which of the following is an example of a catabolic reaction? Protein synthesis, DNA replication, glycolysis, formation of starch. Catabolic reactions break down molecules, glycolysis breaks glucose. Protein synthesis, DNA replication, and starch formation are anabolic, building. Question 7. A change in the DNA sequence that results in a premature stop codon is called a what mutation? Missense, nonsense, silent, frameshift. Nonsense mutation introduces a premature stop codon, truncating protein. Missense changes one amino acid, silent changes no amino acid, frameshift alters reading frame. Question 8. The 2 strands of DNA are held together by which type of bond between complementary bases? Covalent bonds, ionic bonds, hydrogen bonds, peptide bonds. Hydrogen bonds connect complementary bases, AT2, GC3. Covalent bonds form sugar phosphate backbone, ionic occur in side chains, peptide bonds link amino acids. Question 9. Which phase of the cell cycle is not a part of interphase? G1, SG2, mitosis. Mitosis, M phase, is cell division, not interphase. Interphase includes G1, S, and G2 phases for growth and DNA replication. Question 10. The organelle that contains Christie and a matrix and is the site of the Krebs cycle and electron transport chain is the what? Chloroplast, mitochondrion, nucleus, ribosome. Mitochondria have cristae, inner membrane folds, and matrix, Krebs cycle site. Chloroplasts do photosynthesis, nucleus holds DNA, ribosomes synthesize protein. Question 11. In which direction does DNA synthesis occur during replication? 3 foot to 5 only. 5 foot to 3 only. Both directions simultaneously. Randomly. DNA polymerase synthesizes new DNA only in the 5 foot to 3 direction. The leading strand is continuous, lagging strand has Okazaki fragments, but direction remains 5-3. Question 12. Which of the following is a product of the light-dependent reactions of photosynthesis? Glucose, carbon dioxide, oxygen and ADP, water. Light-dependent reactions produce oxygen, from water splitting, ATP, and NADPH. Glucose is made in Calvin cycle, CO2 is used, water is a reactant. Question 13. A solution with a pH of 3 is considered what compared to a solution with a pH of 6? 3 times more basic. 3 times more acidic. 1,000 times more acidic. 1,000 times more basic. pH is logarithmic, base 10. pH 3 has H plus equals 10 to the power of negative 3, pH 6 has H plus equals 10 to the power of negative 6, so pH 3 is 1,000 times more acidic, 10 cubed. Question 14. The process of copying a segment of DNA into RNA is called what? Translation, replication, transcription, transformation. Transcription synthesizes RNA from DNA template. Translation makes protein from mRNA, replication copies DNA, transformation uptakes foreign DNA. Question 15. Which cellular structure is made of microtubules and functions in cell division by forming the spindle fibers? Centriole, ribosome, lysosome, nucleolus. Centrioles organize microtubules to form spindle fibers during division. Ribosomes synthesize protein, lysosomes digest, nucleolus makes ribosomes. Question 16. When a cell secretes hormones or neurotransmitters via vesicle fusion with the plasma membrane, this is called endocytosis, phagocytosis, exocytosis, pinocytosis. Exocytosis fuses vesicles with membrane to release contents. Endocytosis takes in, phagocytosis engulfs large particles, pinocytosis engulfs fluids. Question 17. In Mendelian genetics, an individual with 2 identical alleles for a trait is described as what? Heterozygous, homozygous, hemizygous, dominant. Homozygous has 2 identical alleles, AA or AA. Heterozygous has different alleles, AA, hemizygous 1 copy, males for X-linked, dominant expresses over recessive. Question 18. Which organelle is abundant in liver and kidney cells due to its role in detoxification and lipid metabolism? Rough ER, smooth ER, Golgi apparatus, lysosome. Smooth ER is abundant in liver/kidney for detoxification and lipid metabolism. Rough ER makes proteins, Golgi packages, lysosomes digest. Question 19. The sugar present in DNA molecules is deoxyribose, while the sugar in RNA molecules is what? Glucose, sucrose, ribose, fructose. RNA contains ribose, has OH on 2' carbon. DNA contains deoxyribose, missing that oxygen. Glucose, sucrose, fructose are metabolic sugars, not nucleic acid sugars. Question 20. A recessive allele is usually expressed in the phenotype only when present in which condition? 1 copy, heterozygous. 2 copies, homozygous recessive. On the Y chromosome only. In the presence of a dominant allele. Recessive alleles require 2 copies, homozygous recessive, for expression. Heterozygotes have dominant masking, Y-linked follows different patterns.